So I'll go ahead and introduce you now. So Donn Viviani is a professor of oceanography at Leeward Community College, and he earned his PhD in oceanography from the University of Hawaii at Mānoa in 2016. His PhD research examined the partitioning of primary production between particulate and dissolved phases, rates of bacterial production, and how these rates varied with season, time, depth, light, nutrients, and pH. During his graduate studies and postdoc, he spent 364 days at sea on 65 research cruises. Since he was hired by Leeward Community College in 2018, Donn has taught 46 classes, supervised undergraduate student research, and spent several years constituting 1/3 of Leeward CC's change agent team in the SAGE 2YC project. This is a nationwide project from the National Association of Geoscience Teachers intended to stimulate improved STEM teaching and broader participation at the community college level. And since 2021, Donn has been the Leeward CC team lead for the UH Red Hill Task Force, coordinating the collection of tap water samples.
Okay, thanks. Normally when I'm teaching, I have, there's always a student who can be counted on to tell me if I've got the screen share right. And so I don't have that right now. Okay, so today I'm going to be talking about student-focused coastal research that we're doing in Pu'uloa or in the Pu'uloa area at Leeward Community College. And part of my motivation here is to make you all a little jealous of all the fun we're having with the Pu'uloa plankton parties, because there's no party like a Pu'uloa plankton party. So just as a general outline, I'm going to give some introductions to the talk. I'm going to give the motivation. I'm going to talk a little bit about the history of Leeward Community College in relationship to Pu'uloa and coastal waters. And then I'm going to talk about some different eras of Leeward coastal marine science investigation, and give some specifics, talk about some of our preliminary results, some of our projects, and then talk a little bit about future directions and opportunities. But for those of you that are regular WRRC seminar attendees or have worked with WRRC, you probably know me best from Red Hill stuff. So I thought I should give you a quick introduction. I'm, as Carrie said, I'm the professor of oceanography at Leeward Community College. I've spent a lot of time as an educator, not just as a community college professor. I've taught 7th grade science. I was sort of the utility person for a charter school. I've worked in museums and taught experiential education. You can see a newspaper here from last century where I'm showing a small child a box turtle. So I have a lot of experience as a teacher. And then in 2006, I went to graduate school at Station Aloha, which is 60 miles north of here. It's a long-term oligotrophic habitat assessment. It's designed to sort of take the temperature, the long-term temperature of the North Pacific subtropical gyre. I didn't really go to graduate school here. I just spent a lot of time here. The North Pacific subtropical gyre is the biggest biome on Earth. It's the biggest ecosystem on Earth. That stretches across much of the subtropical Pacific. And this station was set up in 1988 to sort of track what's going on in the ocean. I spent a lot of time here, and I learned a lot, not just in classes at Mānoa, but also going out to sea repeatedly with people who were used to going there, people who've been out to this station 150 times, 120 times. When you're that familiar with an ecosystem, you sort of expect, you know what to expect, you know what not to expect. If you're looking at sort of the depth structure of the salinity or of the temperature, you might see something that doesn't look right. You have a really good feel for what to expect and what not to expect. So I learned a lot just from being with the technicians and scientists of this time series, and it really sort of shaped how I thought about science. I spent a lot of time in graduate school. If this was a movie, we'd have a training montage. So this is my training montage. Spent a lot of time at sea, a lot of time doing experiments, a lot of time filtering water. It's sort of unavoidable. And okay, so you spent a lot of time at sea. What were you doing? Well, primarily I was breaking records. I set the record for the most participation in Hawaii Ocean Time Series cruises by a graduate student. That's actually probably not something I should be proud of. I could have gotten done a lot faster if I hadn't spent so much time at sea. So what I was really interested in was things like, how do phytoplankton production, so photosynthetic production and respiration how well do they track in space and time? Much of the ocean is really nutrient limited. It's something that I stress to my classes. And there's a place across as you cross the equator, there's an increase in the nutrient field. There's more available nutrients. So we expect that photosynthesis will be stimulated by this, that you'll see an increase in primary production. And so I was on a transect cruise across from Fiji to Honolulu across the equator. And the question was, if primary production ramps up, does respiration change, or does respiration sort of stay constant as you move south to north? And from our work, it turned out respiration didn't actually change that much, especially compared to primary production. So I was sort of interested in this balance between production and respiration. And then when I moved on to work on my PhD, as Carrie said, I was interested in this partitioning of newly produced photosynthetic material between the particulate and the dissolved pool. Phytoplankton cells are really leaky. And so if you think of a phytoplankton cell as being like a tea bag, it's probably surrounded by this sort of cloud of stuff that it's released. And if you're looking at newly produced photosynthetic material, how much of it is in the particles, is in the cells, is in the stuff you could pick up if you had a small enough tweezer, and how much of it is in sort of just dissolved out into the seawater, because that's the stuff that's sort of immediately available to bacteria. And so I was interested in that, in looking at that. And to get at this, I went out on these Hawaii Ocean Time Series cruises, 10 a year, over more years than I want to admit. And so I was able to sort of look at primary production, sort of these nested scales of variability from daily scale. How much does the primary production in this one particular patch of ocean, how much does that vary on a daily scale? How much does that vary, at a weekly scale, at a monthly scale, at a seasonal scale? Daily scale was in the summer, because that was when we had the cruise where we spent days and days and days at sea. There's at least one person in the chat I saw who was out there on this cruise also. So this is a long time to be there. But there's some variability in this, and I looked also at the bacterial growth as well, right? How much does bacterial growth change on daily scales over the course of the summer? How does it ramp up? Is it sort of monotonically increasing? How variable is it over sort of monthly timescales? And I know that this is not really the kind of thing that you probably is normally talked about at WRRC seminars. But the response of anyway, the variability is important, and that was one of the things that I was really interested in, even to the point of looking at sort of hourly, or not hourly, but on a daily scale, what happens to bacterial production. And there's some bacteria that are sort of that are just heterotrophs, and there's some bacteria that are photoheterotrophs. They use light energy as well. And so when you look at that, or at least when you look at that in the summer, it turns out that the light, the production, the growth of the light using heterotrophs peaks right about when you'd expect, in the middle of the day, and the strict heterotrophs, or the ones that are growing heterotrophically, seems to peak in the early evening. And so there was sort of this offset in time between the bacteria that are photoheterotrophic and the bacteria that are just heterotrophic. And this was sort of a constant theme, was that you had sort of these offsets in time between different types of events. And so I spent a lot of time out at sea looking at these sort of relationships between production and respiration, production and growth. And so naturally, the stuff that I've been working on recently is freshwater. Working on, I collected samples for a water isotopes project once I started at Leeward. And also I've been working with the Red Hill Task Force. This is still from a training video that we put together to sort of look for volunteers to go out and collect tap water samples from the community. And at first that might seem a little weird that I'm working in fresh water and tap water, which is not at all like the ocean. But first of all, the Red Hill, a lot of the Red Hill response was kicked off by a student email to a colleague of mine. Hey, my tap water smells like gasoline. Is there anybody who can test it? And even though I did my PhD on oceanic waters, the question of what's in water is really compelling and interesting, and especially because I live here, and so there's the scientific interest. There's also the sort of self-interest of what's going on in our water, and so I've been working on a number of different projects at Leeward, and I've sort of put them together here with the names of many of the people that I've been collaborating with. The city water project that I mentioned, where we were collecting these water samples that were analyzed at in Utah for the stable isotopes. I've been working on other projects, and the other thing, though, and if you look at sort of this Venn diagram showing the different projects that I'll sort of get into more later, and also who I've been working with, there's something that I want to make sure I point out, and that's that students are working on all of these, and that's sort of the central point, is that I have all these projects that are engaging and training students and teaching them skills they're learning from doing this. So I'm going to talk a little bit about these projects, and I should point out that a lot of the data that's collected that I'm going to show, the pictures that I'm going to show, were taken and collected by my collaborators and my students, and so they've done really wonderful work, and the questionable visual choices and design choices are all mine, but the really nice pictures are all theirs. So I want to make that really clear. Okay, so the motivation for this talk is I really want to highlight Leeward CC students. I think Leeward CC students are really excellent, and I don't just think that because I teach them. I think that because people tell me that Leeward students are really, really top notch. They're really well prepared. That they're really valuable. When they transfer to four-year schools, when they're in classes, when they're working in labs, they're really good. And I hear that a lot. I also want to point out the incredible potential of Pu'uloa, of Pearl Harbor, of the Three Lochs, of Pearl Harbor for research, for interesting science. It is, I think, very understudied. When I first started working with students in Pu'uloa, and we started looking for, that's what you do when you start working in a new system, you start looking at the literature, and there isn't actually that much published about Pu'uloa. There isn't that much known. And so there's a lot of really interesting work, I think, that can be done. I'm also here to spread the word for collaborators, for partners, future advisors for my wonderful students who I hope will all transfer to four-year schools and are going to be looking for more research experiences, are going to be looking for advisors, maybe going to grad school someday. And so I want to, let people know that these wonderful students are out there. If people have ideas for more funding, I'd be delighted to hear that. And I also want to sort of show off how much fun we have as Microscope Shark can show you. This is a picture from a costume party that we had a couple of years ago, and people came in costume, and they did some research in costume, and then they had a good time. Okay. So Leeward Community College typically is the highest enrollment community college. We serve the largest number of Native Hawaiian students. I think the highest number of military and veteran students among the community colleges. I don't know if we serve the most, but we have a large number of Filipino students. We have a lot of first-generation students, and we draw from basically across the Ewa and Waianae areas. This is a breakdown of recent sort of student diversity, showing the self-reported ethnic breakdown of our students. And so the point is, if you are interested in recruiting and attracting local students, Native Hawaiian students, first-generation students, you can find them at Leeward. That's where a lot of them are, and this is the chance to grab them and attract them to STEM and to other interesting disciplines. Okay, so Leeward not only Leeward has a really strong history with the ocean sciences, with the marine sciences. This semester, if you look at enrollment in Ocean 201, that's Science of the Sea, that's the basic oceanography course taught across the system. A third of UH system students that are taking Ocean 201 are taking it at Leeward. That's just on the Oahu campuses, but that's a third of the total Ocean 201 students on Oahu are taking it at Leeward. That's shown here in this pie chart. This is Leeward is in green. Now, yes, this semester Mānoa is teaching more students Ocean 201, but Mānoa also has like 6 times our undergraduate enrollment, right? Like typically, Leeward is always either teaches the most or the second most Ocean 201 students, and it's always above 20%. And in fact, 39% of students taking the Ocean 201 laboratory this semester are at Leeward. And that's not just this semester, and as much as I'd like to claim that it's all due to my wonderful teaching, it actually isn't down to that at all. There's been a history of strong 201 enrollment. These classes continually fill. In fact, I'm told that there were 2 full-time oceanography instructors back in the 70s, and one of them told me that himself. This lower picture on the right is a picture of Donn Plim, who was oceanography instructor at Leeward Community College in the 70s and 80s. And Don had been retired for a while, and he heard there was a new instructor, and so he showed up at my door one day. This man walks into my room and demands to know if I'm the new instructor, and we started talking. And so I got sort of a feel for the history of Leeward, not just the history of Leeward, but the history of the marine sciences, of oceanography at Leeward, and all the stuff that they were doing in the past. Because it turns out that Don and students were doing coastal science, we're doing marine science, we're doing oceanography in the 70s and 80s. This is a sketch of the study sites in the streams that feed Pu'uloa from the work that Don and his students were doing in the 70s and 80s. And so here, I'll orient you if you're not that familiar. This is West Loch. This is Middle Loch, where the mothballed ships are stored. You can see these mothballed ships from the Leeward campus. This is, and this actually hurts a little bit, I've circled in red the structure that says LCC dock, Leeward Community College dock. We no longer have a dock. We don't have a boat anymore. That's sort of that's a someday maybe. But the point is, Leeward is right up here. And that's, I think, one reason that we're really well positioned to do this work. Over here is Eash Loch. This is there's Blaisdell Park is right here. This is Waimalu Stream. Here's Ford Island. And so these students were measuring and monitoring stream water quality and other parameters in the 70s and 80s. They were also actually going out into Pu'uloa. This is another map put together by Donn Plim, showing the stations that they would occupy in the 70s and 80s, you know, 6, 7 times a year, maybe less, you know, but the point is, this is data that is probably unique, and it's available online. I also have copies of it. You're more than welcome to it. I'd be delighted to share it with you. And this is really nice data because students can use it. When I first started at Leeward, I had a Mānoa student who was looking for a mentor for her OEV undergraduate research fellowship, and she wanted some data to play with to work on coding and building models and data visualization. And we had this wonderful data from Pearl Harbor, and so we worked on this a little bit. And so, you know, so she put together sort of this. She started looking at things like, what's the dissolved oxygen like in Pearl Harbor? You know, I have this. You know, I kind of feel like there's probably some really interesting oxygen dynamics in the harbor. You know, maybe there's some suboxic or anoxic zones, and she started going through the data and looking for these stations, what the oxygen is like, have there been temperature changes and so on. So, and here she's graphed oxygen against temperature. But the point is, we have this data, and so, you know, when we're sort of looking at can we go out and do some research now, we can sort of use that past data that was gathered by students to inform where we want to go now. So again, I've got 3 stations circled in green here, one right here in the middle of West Loch, one here at the mouth of Waimalu Stream, and one all the way here at the entrance to the estuary. And, you know, for example, when I pull data from Station 6 here in West Loch, you know, from 1973 to 1984, you know, we've got measurements of oxygen at the surface, basically, and at 5 meters depth. And at 5 meters depth, sometimes there's almost no measurable oxygen in West Loch. And that's really interesting. We can't get out there right now, you know, hopefully we can, but it'd be really cool to see. Well, there's all kinds of things you could ask about that, right? Is that a ephemeral thing, or is it always really suboxic, right? Is there really interesting stuff going on? You know, I probably Anyway, there's probably really interesting stuff going on there. So I'd love to get out into Pearl Harbor and do this kind of work. And you know, that's the hope. I've also plotted Secchi depth, and a Secchi disc is a piece of 16th century technology. It's a plastic disc painted white and black with a line on it, and you lower it into the ocean. And when you can't see it anymore, that's the depth of light penetration. So there's the region in which you can have photosynthesis. Everything below that is purely respiration. And so here you can see that there's over this 11-year stretch with sparse sampling, sure, but over this 11-year stretch, generally the Secchi depth, the distance of light penetration into Pearl Harbor is 1.5 meters, between a half meter and 1.5 meters, maybe 2 meters, except for this one particular event where it was really clear, right? And that's really interesting, right? Like, what kind of differences do we see in the stuff that's blocking light, right? Is it particulate? Is it phytoplankton blooms? Are there lots of diatoms? What is going on here that sort of leads to this? So there's a lot of really interesting, stuff that can be done. So when I was starting at Leeward, there was this grant that I started working with that had a little money for students to do research projects. It's called Kiloaina Science of Place. And I had already seen this data of Donn Viviani, and I decided to use this sort of as a guide to where I should have students sample. And so I picked basically the mouth of West stream, because we had some data from there, as close as we could get to the outflow of the estuary. And also we couldn't get out to the middle of West Loch, but we picked a place in West Loch that we could easily access, so we could sort of try to get some measurements. And so student research sampling sites were chosen based on this past data, and also on whether or not they were accessible. And so we found some accessible spots, and we had a bunch of students sampling at Blaisdell Park, at the mouth of West stream, right here over this over the bicycle bridge, at Iroquois Point down here near the entrance of the mouth of the estuary, and then over here at Kapapapuhi Point Park, which is a park, and so it's freely accessible. And so here are students sampling at all three of those locations. I should mention that there are 3 students who got involved in Kiloaina and have stayed involved with this. Jessica, Marlon, and Kelsey, who were part of this initial sampling and have stayed around for a while doing other projects with us. So we had this data collection, and students really struggling with the big issues, right? Like, why plankton and why should we care? And really, because it's the base of the food chain. Like, if you care about fish, the larval fish are part of the plankton, and the larval fish are part of the plankton, which means they're in that plankton fraction. They're being eaten by other plankton. They're eating other plankton, and then when they grow up, they're still being supported by plankton, because that's the base of the food chain. Whether you're talking about the phytoplankton that are photosynthesizing, or whether you're talking about the zooplankton, the animal plankton that are eating them, right? That is the base of your food chain. So if you care about fish, you should care about plankton. So students were looking at plankton, and the other thing, and I think part of what's made this a success, is the plankton are really cool to look at. So these are some pictures taken by collaborators and students, and we have everything from a copepod carrying eggs to like a wider view of, it looks like copepods and crustacean nauplii, probably copepod nauplii juveniles, and some barnacle larvae. Again, barnacles are crazy because they're crustaceans. So even though they seem like, even though they, as adults, adopt this set in place lifestyle, they're really highly mobile as juveniles. So we've got medusae, there's diatoms here and here and here and here, all different types of diatoms, photosynthetic phytoplankton that live in glass houses, marine snail, polychaete worm, and Noctiluca scintillans, also called sea sparkle. This is a bioluminescent zooplankton that we pretty regularly find in West Loch. I've had a couple students try to look at whether this particular Noctiluca is bioluminescent. Those are sort of quick, easy experiments. So I think there's a lot to be done there, but they're really cool looking. Most of them are really cool looking, and also these Kiloaina students then started to build up an idea of which plankton they expect to find when. So this is results from multiple semesters of Kiloina, and it's a heat map showing when particular types of plankton are absent, are present, light gray, or are sort of dominant, like that's the majority thing you're seeing, right? And so obviously this isn't quantitative, this is qualitative, but even so, you start to get a feel for what you expect to see, just like when I was going to see with the HOT program. Students, myself, can say, all right, we're going to go to Kapapapuhi Point, we're going to pull a net, we're going to look under the microscope, and here's what I expect to see. I expect to see diatoms, I expect to see copepods, I expect to see these barnacle and copepod nauplii. We might see Noctiluca. We're probably not going to see Keating gnats, although we might. So you start to get a feel for what's there. Not only do you start to build up an expectation for what might be there, so you can sort of get a feel for what's there. You can also, as you have these pictures, you can start to generate a guide, so that if you have a new student, if you have a class looking at plankton, you have this nice guide to say these are the things that you will commonly see that you can sort of use to identify. Also, by going repeatedly and taking some other measurements, you can start to think about the environmental context. What is the salinity of these three sites? Again, we have a stream mouth where on one of the sampling occasions, there had been a big rainstorm, and so basically it was fresh water. Iroquois Point, which is the entrance to Pu'uloa, to Pearl Harbor, had the highest salinity, and then Kapapapuhi was sort of always salty. But you can also start saying, all right, well, we have these we have this sort of presence absence data for plankton. Can we map that to any environmental variables? So like people often talk about how particular types of jellyfish, say, are tied to moon phases, right? So when we look at medusae, for example, when we find medusae, does that correlate to the moon phases? My colleague Anushka Fauci put together a sort of a richness index for the plankton data, and we could sort of look and see at these sites, does richness sort of vary with moon phase? If you look at particular types of plankton, are they more common at certain tides? So, for example, I pointed out this Noctiluca before, this sea sparkle. And when Noctiluca is abundant, it's really abundant. You hold up a container holding the net toe, and you can actually sort of see a purple pink haze inside it because there are so many of these things. Does that relate to the tide, right? So I sort of put together an index of tide. Ebb is negative, flood is positive, and trying to see, when Noctiluca is really abundant. Does that correlate with any particular tide? And this is the kind of thing we can start to investigate, like how does the environment relate to the presence of these organisms? From this early Kiloina work, it looked like richness was well correlated to salinity, and I don't think this has to do with anything about the salt. I think this is sort of more a reflection of sort of ocean input, right? Because the tightest correlation was at Iroquois Point, where you have that connection to the sea. And so probably, when you're getting inflow from the ocean, it's slightly more saline, the salinity is higher, and so you probably have higher diversity. But again, this is the kind of thing you can start to do, and you can start to put together these expectations. You can start to test questions. And you can put together, as I said, a species guide and list, so that not only do you have that for when the students are going out, when they're training new students, if you're keeping this going, you also can use it for your classes. You can print out a bunch of these, and when you have, for example, a lab on plankton, you can do an ETO, you can bring it in, you can say to the students, all right, I want you to look through and try to identify these things, and these are the things you're most likely to see. This is what they look like. And there are obviously available books on identifying plankton and phytoplankton and zooplankton and so on. But having this kind of very specific guide where we know these things are in Pu'uloa because this is what we normally saw, that's really valuable, I think. And so that's been really useful. Okay. So we had this great project. Students really enjoyed it. We got some really interesting data out of it. And then the funding ended. And so, all right, so then I was sitting there thinking, all right, there is still the possibility to fund student research. The Bridge to Baccalaureate program is a community college-wide program that gives stipends to students every semester who are working with professors. Through my collaborator, Barb Bruno, there were grants that were available through the GeoPath program, headed up first by John Rand and then by Hervé Collin. There was money available. And we had these really capable and interested Leeward students, but it's typical to get money for supplies, right? Student stipends are easy. And also, I didn't necessarily have that many ideas, so I wasn't sure what I could do to sort of guide students to opportunities. So I started, first of all, directing them to opportunities that were already existing. There's been this wonderful collaboration between folks at Mānoa and KCC, Oceanic Community College, and Oregon State, looking at the Sumida Farm. Sumida is a watercress farm. It's been in operation for over a century, I think. It's right in the middle of Pearl City. You're driving along underneath the rail, and there's, you're passing, restaurants and banks, and then there's this watercress farm, and it's full of birds, and egrets, and it's gorgeous. And the Sumida family has really been very generous in sort of letting students use it as a place to do research, to get experience. They're interested in water quality issues, but they've been very good about saying, you have a student who wants to study birds, come in and survey the birds. You have a student who wants to look at inverts, come on in. So I've had a student, Nate Chen's been looking at invert diversity and comparing Sumida Farms, the sort of deepest place where the water pools, watercress, right? There's a lot of water there. So he's comparing the plankton of this region here in Sumida Farms to the outflow of this stream, which is not far from our Blaisdell site in the Kilauea days. And so there's these opportunities to get involved and direct students to something they're interested in. Nate is interested in looking at invertebrates. And so this is a chance to go out into the field, and he's recruited some of his fellow student researchers like Azrael to go with him and help collect these samples. But again, I was sort of stuck in this space where where I could find money to fund students, and there were really good students, but I needed more ideas. And then I realized that I actually had space and I had equipment. I have microscopes, I have space, right? And that's a really valuable thing. And then I started talking to people who had samples that they maybe didn't have the time to process, or they had the time, but maybe they were a PhD student, and so it would be helpful for them to have assistance in working on these samples. So these together led to these collaborative student-centered research experiences that I'm really excited about, and that we've been working on at Leeward. So Cassie Lyons is a graduate student at HIMB at the Hawaii Institute of Marine Biology, and for her PhD, she is looking at fish eggs and fish larvae from Kaneohe Bay, and she's going to compare that to a similar survey that was done in the 1970s. And she's also going to do some molecular work looking at these fish larvae. And this is really interesting, right? Like what has changed, what hasn't changed, what do we see there? And so students have been trained to come in, and first of all, we were just separating out, the eggs and the larvae from the masses and masses and masses of chaetognats or arrowworms that you typically find in a Kaneohe Bay sample. Kaneohe Bay is full of chaetognats, and if you're looking for something to keep you awake at night, Google an image of a chaetognat's jaws. But this was the opportunity for students to come in. They learned how to differentiate the different larval fish, separate out interesting things like octopus. You can see a flapjack octopus here, right? Eel eggs. Here's an eel egg picked up by tweezers. And then by partnering with my colleague Helmut Kay next door in microbiology, start to work on molecular techniques, have some students learn not just microscope, not just identifying, but also molecular techniques. So I have this collaboration with Cassie. I've also been working with Don Kobayashi from NOAA, and who had this wonderful treasure trove of plankton samples from the twilight zone, from deeper in the ocean below that light layer where the light goes from enough to photosynthesize to enough to see by. And Don and these NOAA scientists are interested in commercially important species, and so he brings the samples in, and the students can go through them, pick them out, learn to identify them, and Don brings other scientists from NOAA who come in and spend time with the students. They share their expertise. Some of them are walking encyclopedias of larval fish. And so the students learn that. They get to see that in action. They get to see scientists knowing what's going on. And they also get to see scientists being unsure what something is. I'm not sure what that is. What do you think that is? I'm not sure. They get to talk to grad students who are from Hawaii who come by and sort of get a feel for what that pathway looks like, right? What is it? What is it like to be a graduate student? What do you have to do? And so this is really valuable. You also get some of this interaction between different students. A student has applied for a summer REU and got it, and the next year they can talk to the other students about what they did to get it, right? And so it's this really wonderful opportunity for students to come in and have this really great opportunity, because it's also a communal thing. It's not just one student coming in, firing up the microscope and getting going. The students come in a group, they work together, and they have fun, I think. At least they seem to. It can be hard to identify some of the smaller fish, some of the smaller larval samples, or some of the smaller fish samples, but some of them are a little easier to pick out. You can see some of those here. This is probably the worst image in the slideshow, and this is the one I took. But it's a really cool black dragonfish, and its eyeballs are all the way out on these stalks. There's just these wonderful, bizarre looking things in the plankton samples. So we have this wonderful sort of student-centered opportunity where students are interacting with scientists, they're interacting with each other, they're learning things, and they're using samples that actually mean something, that are going into a project, that are answering specific questions. But I did want to sort of have a field component as well. And so I kept sort of talking about how I needed to restart the time series. And last summer, one of the students, MJ, asked if she could sort of plan things out. And so, because, I really did feel like there's this importance to being able to having some fieldwork experience, to getting out in the field, to learning to sample, problem solving, learning a bunch of different skills. If you're doing microscopy on fish larvae, you'll learn to identify the fish larvae, and you'll get good with the microscope. But if you're doing sampling in the environment, you need to know a bunch of different things. And if you're not an expert in them, you at least need to know them. And I can see Pu'uloa from my classroom door, right? I can see Middle Loch, but even so, I can see it from my classroom door. And I've gotten really into plankton. And there's this important public service aspect that, Red Hill also kind of crystallized for me, right? I mean, you have this leak of this jet fuel into people's water. But when I was going out in the community and talking to people, they were also pointing out there are Superfund sites, in Pearl City Peninsula, for example, right? And this stuff is eventually going to end up in Pu'uloa. And, so we don't know how polluted Pu'uloa is. There are all these signs that say, contaminated fish and shellfish for your health, do not eat. But just about every time I've gone there to sample or to take classes, I see people fishing. Now, maybe they're just sport fishing, catch and release, but maybe not. And so it'd be really nice to have more information about the area. I'm not necessarily, I'm not going to be able to do the toxics analysis myself, but I can provide Leeward can maybe provide some of the environmental context, some of the species diversity information that could contribute to this. And if I can get this time series going again, you can sort of train students and train them into a project that they feel like they're part of something that's long lasting. Newer students can learn from the older students, and students often learn better from other students than they do from their professors. In a way, I think, because I was so heavily influenced by the Hawaii Ocean Time series, I was hoping to recreate a little bit of that in the Pu'uloa Time series. Get some group to group and peer to peer learning experiences. And also, I think this facilitates partnerships. If you have a question about medusae, or you have a question about diatoms, or you are interested in bacterial films, if something is known about a particular site, right? If there's a time series that is collecting sort of this environmental data, it's really easy for you, or I think it's really rewarding to say, all right, I have this project, I'm going to hook it onto the time series here, collect a year's worth of data, and then put an end to it. And I have a year's worth of ancillary data and also, data from the past and hopefully data from the future. So I think it's a really valuable thing. And so my hope was that I could sort of turn Kapapapuhi Point Park into an adjunct to my classroom, use it for teaching and research. In the course catalog, a field trip is required for my Ocean 201 Science and Sea class. And so I would try to take students to Kapapapuhi Point Park, sort of bring some microscopes and pull a net and let them look at the stuff that was under there. When I'm lucky, there's a Noctiluca bloom, not, like up in the upper right, but sometimes there's this Noctiluca bloom. And so, it's this really wonderful opportunity that's 15 minutes away from my classroom, maybe, right? Maybe 13 if there's not that much traffic. So as I said, MJ started to spin up a renewed time series. And the difference was that we were going to sample both sides of the point. So this is West Loch. Here's Kapapapuhi Point. It's a park. And the interesting thing is this north side has had the mangroves cleared from it by uloa. But the south side, the mangroves are still present. So you have a cleared north side and a south side that's still covered in mangroves. And so that's really interesting, right? Because that sort of is a built-in experiment, right? What is the difference? Or what differences do we see between the water on this south side where the mangroves have been cleared and on the north side where the mangroves are still present? And if the mangroves get cleared in the future, that's another really interesting experiment, right? Like here's what it looked like up until the mangroves got cleared. What happens once the mangroves get cleared? Like what does that disturbance do, right? What can we find out? So I think it's this really interesting opportunity it's this really interesting opportunity it's this really interesting opportunity that you don't often you couldn't plan, or it'd be really hard to plan. So MJ and Andrew Garma and Azrael Tan have been going regularly, maybe other students. I think Jessica Kino has been involved in this, have been going down to take samples from Kapapapuhi Point over since the past summer. And so, for example, when they look at the temperature, they started by being really ambitious and looking at both high tide and low tide. And so you can sort of see that at low tide for much of the summer, the mangrove side was a little bit warmer than the cleared side. At high tide, there wasn't much difference. Didn't seem to be a pattern. They were looking at salinity at low tide compared to the cleared side and the mangrove side. And high tide, seems to be a bigger difference between salinity between the cleared side and the mangrove side at low tide compared to the high tide. And I haven't done any statistics on this because I don't think we really have enough points to say. But the point is that we're starting to build up this picture of what these two sites look like. And you might wonder what plankton we're finding. And we've been collecting plankton samples. They are preserved in ethanol, and we're going to start trying to identify them. We're starting to work on that now. We've been focused on one of the other projects a little more, but this is something that we're looking forward to moving on, because again, right, we have a plan for making this an ongoing thing. We're going to do these plankton comparisons, tie in larval fish with because we have collaborators who are interested in larval fish, right? Cassie is interested in larval fish at Pu'uloa, right? What do we know about diatoms, right? Because we have this information about Kapapapuhi Point. We have this idea of what we expect to find all the time, what we expect to find regularly, what we'll occasionally find, what we don't expect to find, right? And we're starting to collect these other water parameters that'll help us sort of establish the context. There may be other things that we fold into this, and maybe there are other projects. Maybe your project, right? Maybe you have a grad student that wants to test out a method. You could go to Krause Pond on the UH campus, or you could come to Kapapapuhi Point, meet some undergraduates, get out and test out a method in the field. Maybe you have some analysis you want to fine tune, and you just need to collect some water a couple of times over the next year to just to get it dialed in. Maybe you've got a piece of equipment that you really want to use, or you really should use, and you don't really have a place to use it. Come to Kapapapuhi Point. We'd be happy to go with you. Maybe this is your chance to get your boots out and do some field work, right? So I really want to extend that as an offer, as an invitation, right? That's my main motivation for giving this seminar is to let folks know that we're doing this thing that I think is really cool. You can come out, you can get some field work in, you can meet some wonderful students who you can then I've been instructed not to use the word poach. You can meet some wonderful students that you can recruit, that can come and do great things in your lab. And here's where I want to shout out the students. And I've divided into a bunch of different research eras, right? Like the Kiloaina era, and then I had a couple of students in the summer after Kiloaina, and then I had a few students working on Red Hill, and then what I like to think of as the Plankton Party era, this era of doing the fish work, the larval fish work, and the Pu'uloa time series. There are a few students who have been here through a number of eras from Kiloaina on, and students in bold are still working with us, are still there, and for many of the students that aren't still working with us, it's because they're now working for the Department of Health, right? They went through Leeward, they transferred to Mānoa, and they got a job at the Department of Health, or they are working with, for example, Aurora, and she did not poach my students. I'm happy that she recruited them. But the point here is actually I am happy that she recruited them, because they're going to get an awful lot out of working with her or working with professors at Mānoa. We hope to give them a lot, and then when they move on to a four-year school, we hope that they will get even more from working with people there. So we have these great students. They have a lot of fun. This is one of those students who was working the program was an early college student, meaning that he was an undergraduate student taking college classes. And so when he graduated high school, the other students had a little party. You could see he has liaison, and I don't. For insurance purposes, I want to stress they did not throw him up in the air. But the point is, the students, I think, really have a lot of fun. And so when I go back to this sort of messy diagram, we have all these really cool things going on, and some of my collaborators are involved in a couple of these different projects, but we have all of these wonderful students that are, I think, getting an awful lot out of these experiences. So I just want to close here. Again, shouting out the research students who are doing a wonderful job. And I want to say that, like, we're looking for collaborators, we're looking for partners, we're looking for future advisors for these great students. Like, recruit them into your lab and teach them wonderful things, and they will do great work. And I know that because they do great work at Leeward. We're always looking for new students. But if you want to recruit top-notch Leeward students to your lab, if you have an undergraduate research opportunity, like, we are recruiting 7 to 10. We have a consistent group of 7 to 10 students a semester, and we're recruiting all the time. As students move on to new things, we are recruiting more behind them, and I think that there's really a lot of research potential in this Pu'uloa time series. So, come join us. And if you are interested in any of this, please contact me. My email is on the slide, and that's all I have today. And I'm happy to take any questions that you might have.
All right, thanks so much, Donn. Really awesome talk, really awesome to hear about the work that your students have been doing. I think it's time to turn it over to the chat. So please enter any questions that you have. I hope that this opened up opportunities for future collaboration. Oh, Aurora.
The talk was given at such a high technical level that I don't think no, I'm just kidding. I'm just kidding.
Sorry, I'm on my phone, so chatting with my thumbs is hard. I guess I have a question, because what kind of questions might you be able to ask about urban pollution and its effects on, like, that estuarine ecosystem, given that Superfund site? And this wasn't a planted question, it's one that occurred to me as it's like, what would you look for? Do you have thoughts on that?
Well, I'm going to filibuster for a minute. Earlier in the year, last year, Jessica, Marlon Velasco and Kelsey Yap and I presented a poster at the Ocean Sciences meeting. And we ended up being in a session with Tracy Sutton, who has been sort of running this long-term Gulf of Mexico study looking at the effects of the Deepwater Horizon spill. And one of the really interesting things that came out of that from his perspective is that, there was a huge effect of the oil spill on bigger animals, for want of a better word, but the plankton seemed to recover really quickly, right? And so I think there's this really interesting thing going on in Pearl Harbor, which is that, first of all, because you're not supposed to fish there, it's basically a nature reserve, right? So you have probably lots and lots of fish there. But also it's alleged to be very polluted. And I'm not saying it's not, I'm just saying, that it's repeatedly very polluted. And so, it's really interesting to say, all right, so we've been looking at plankton, and we have this plankton diversity, right? And if diversity crashes, that probably tells you something about the health of the estuary. Because I think there was Tracy saw a short-term plankton crash with the Deepwater Horizon. But I think there's a real opportunity to sort of look at the larval fish, to see what the fish populations are like. And I think, realistically, like things roll downhill, and so there's an opportunity to sort of look at the health of uloa, and that'll tell you something about maybe about the entrainment or the transport of toxic substances upstream, I think. So I think there's opportunities there. I don't have necessarily an answer to your question, but I think it's the kind of thing where you could, if you're regularly going out and you're collecting samples that could tell you something about contamination, then you have a time series and you can say, all right, we didn't see anything until March of 2024, and then we saw a spike in something, and then we didn't see anything. So, was there a big rain event? Was there a release? So I think that could tell us something.
Okay, we have a question in the chat. Actually, I think that I'm going to allow people to raise their hand and unmute if that makes it easier to facilitate discussion. So I'll ask Gordon if he wants to ask his question verbally.
Hey there, can you hear me?
Yeah.
Yep.
Okay, sorry. Yeah, good. Really interesting talk. I'm curious about a bunch of things, but I think for this forum, really, the question is, there's a lot of security at Pearl Harbor, right? And I'm wondering what sort of relationship you've had with the Navy in terms of getting access.
So this is more of a asking forgiveness rather than permission scenario. No, we chose sampling sites that were publicly accessible from the shoreline. We'd love to get out into Pearl Harbor. We had talked about, I think I sent an email to NAVFAC before the pandemic asking if it was possible, and I hadn't heard back. I think it's difficult to get access. I'm hopeful that we can at some point, but I haven't had much discussion with the Navy about. I haven't had discussions with the Navy about this at all. Thanks, Tom. Yeah, Gordon. So I haven't really had much discussion with the Navy, so I don't know how cooperative or not they would be. My sense from talking to other people who've gotten permission to say go out into West Loch on a stand-up paddleboard is that it can be kind of a pain, but that's definitely something to pursue in the future.
Okay, there's another question from Melody. If you wanted to ask it out loud, you can go ahead.
Oh, okay. Yeah, I had a question. I've heard about a project several years ago about the placing of oysters in uloa that was designed to help filter out the oil and jet fuel that was in there. And so I was wondering whether your students may have been involved in that project, or you have been involved or know anything about it, whether there's any success, because I'm concerned with regards to uloa having been the breadbasket of the kingdom. And it would be nice if we could make it such that we could consume the seafood. And you're right, people are fishing there and hiding what they fish because they know it's not supposed to be consumed. But how do you stop that if we still see people throwing nets and things like that?
Okay. So with respect to the first part, I had no. I don't have any connection to the oyster project. What I will say though is that oysters are filter feeders, so they're pulling particles out of the water. So they might be eating some of the small. I'm not sure what size things oysters eat, but they may be eating some of the smaller plankton. But we do sometimes see bivalve larvae in our net toes. So we could look at what type of bivalve that is. I mean, that's probably a molecular analysis, looking at the DNA rather than identifying by eye. But you know, we could see, you know, hey, are these oysters reproducing? And I think that, like, you know, originally at one point I had hoped that we could get a flow cam, which is this automated instrument that you can hook a plankton sample up to, and it'll sort of cycle in front of a camera that is connected to a computer, and it'll sort of automatically identify things after you train it. And so that would be, you know, that would be something I think useful for a project like the Oyster Project, you know, to know like what's in the water. I mean, even from our work, you know, here's what's in the water, in the plankton. Here's the general size classes we see. You know, do we see oyster larvae or not? Right. So that might be really interesting and useful for them. With respect to the fishing issue, I think there are labs. There's a lab at HIMB. I think the Donahue lab is working on sort of the toxic materials in fish, and but you know, one benefit to having a time series at Kapapapui Point, where people often fish, and having a time series that's worked on by local students, is that it becomes a little easier to sort of spread the word out. Like sometimes when I'm walking down there with my buckets and my net and my, you know, other gear, people will stop me and ask what's going on, right? And so I think if we're there regularly, there's an opportunity for dialogue, and we can talk to people about what's out there. And so I can't do anything about people fishing, and I'm, you know, I mean, people are going to do what they're going to do, but I think we have the opportunity to sort of gather some information and maybe to do some education.
Thank you.
Thank you.
So thank you all, and have a happy Friday and good weekend.
Thank you so much.
Thanks, Don.